Alright, this is just a real quick review for tomorrow's test. A couple people asked me if I would record this. So the short answer on the test is going to be something experiment-based. It'll be questions about either photosynthesis, respiration, fermentation, but it'll be situational. So here's an example. It won't be this experiment, but here's a good example. So let's say that I tell you that I have an indicator, and the indicator in an average amount of carbon dioxide is purple. If the levels of carbon dioxide go up, that indicator turns blue, and if the levels of carbon dioxide go down, that indicator turns red. And then I give you situations, and I ask you, what color will the indicator turn? So the first situation I could give you is a plant in the light. So if we look at our first situation here, we have a plant growing in the light, and the question is, what color will the indicator turn? The correct answer is, in this case, the indicator should turn red. The reason why the indicator is going to turn red is because in bright light, the dominant process in this plant is probably going to be photosynthesis. And photosynthesis takes in CO2 and makes sugar and oxygen. So the levels of carbon dioxide should go down, and that would make the indicator turn red. The second situation is a plant in the dark. If you were to just tell me that no photosynthesis is happening, you really only get half credit here. Because technically the correct answer here is that the indicator would turn blue, because although it's true that photosynthesis does not occur in the dark, cell respiration is occurring all the time, in the light and the dark, in every organism. And cell respiration takes glucose and oxygen, breaks it down into CO2 and water, which means the carbon dioxide levels should go up in our second one. In our third one, we have a mouse. The mouse is doing cell respiration and not photosynthesis, because animals can't do photosynthesis. It shouldn't matter whether the mouse is in the light or the dark. The mouse doing the process of cell respiration should make the CO2 go up, and so the mouse, just like the plant in the dark, should turn blue because the levels of carbon dioxide are going up. I could even change it up and put a mouse and a plant. And probably the most logical answer there would be that the indicator might stay purple, because the plant would be doing photosynthesis, assuming there's light. In the dark, that wouldn't be true. And the animal would be doing cell respiration. Plant would technically be doing some cell respiration also, but because CO2 would be made and consumed, we probably wouldn't see a color change. So that's that kind of an experiment is what you should be able to do on the test. I could, instead of CO2, I could say oxygen. Instead of colors, I could say pHs. It could be, there's a lot of variations of what I could do. I could also do something that had to do with fermentation. So keep in mind that fermentation in us, the products we would see would be lactic acid. And if it was yeast, for example, it would be CO2 and ethanol. Those would be the two products of fermentation that we would see. Whereas if oxygen was present, we wouldn't make lactic acid. We would continue and do the whole process of aerobic cell respiration, and our products would be carbon dioxide and water. Yeast and most bacteria, they don't usually use oxygen. Fermentation is a natural pathway for a lot of them. They don't have the ability to use oxygen, so they always make carbon dioxide and ethanol as their products. The other thing that you should know, the other short answer question, will be something like this, and the numbers will obviously be changed, but I'll tell you that you have some number of NADHs and some number of FADH2s, and they go to the electron transport chain, and I say how many total ATP would you be able to make from them. And the way you're going to solve this is every NADH can make 3 ATP, and every FADH2 can make 2 ATP. So we just multiply. 5 times 3 is 15, 2 times 2 is 4, and so the answer in this case would be 19 ATP. But again, it won't be these numbers. Just make sure you memorize that when NADH gets recycled, 3 ATP for every NADH that's recycled, and 2 ATP for each FADH2 that gets recycled, oxidized back to its original form.